So here we're going to talk about what happens when you have anaerobic conditions following glycolysis, which basically means there's no oxygen present, or you're in a red blood cell, for example, and you only can do anaerobic glycolysis. So anaerobic conditions means you don't have oxygen, so you're going to have to do something with your NADH. So your NADH cannot go to the mitochondria, and so you're going to have to transfer those electrons to pyruvate to form lactate. So in this case, pyruvate becomes the final electron acceptor in essence, and it forms lactate, and this is through lactate dehydrogenase. And it's important because you're regenerating NAD, so this can go back to glycolysis. Otherwise, you get everything trapped at NADH. So NAD must be regenerated via lactic acid fermentation. Now again, there's no net change in anaerobic with NAD or NADH. When you convert glucose to pyruvate, you generate 2 NADH, and when you generate pyruvate to lactate, you generate 2 NAD. So it's a cycle that keeps this going. And remember, in glucose, you have a net ATP of 2. So you want to make sure to keep that available so that you can keep making that net 2 ATP. So for anaerobics, you have one glucose molecule that comes in, and you have 2 inorganic phosphates, 2 ADPs, and it results in 2 lactates, 2 ATPs, and water. Lactate and hydrogens are going to be moved to the blood, and this can lead to an increase of lactic acid and lactic acidosis. Anaerobic glycolysis is going to be used by the red blood cells solely, and it also can be used, of course, during muscle when they're having contractions, the eye tissue and other tissues, your medullary cells of your kidneys, for example. So if you're going to rely on anaerobic glycolysis, you're going to have to be much faster at pushing glucose through glycolysis. So you're going to need more of those glycolysis enzymes, and you're going to need more glucose. Now, aerobic versus anaerobic as far as energy yield. And anaerobic, you're only going to be able to generate 2 moles of ATP per mole of glucose. When oxygen is present, you can tap into that NADH, and oxidation of pyruvate alone will yield 12.5 moles of ATP. Your cytosolic NADH will generate 1.25 ATP if you go through that glucose 3-phosphate shuttle, or 2.5 if you go through that malate aspartate shuttle. Altogether, you can get 30 to 32 ATP produced from 1 mole of glucose for aerobic, and only 2 for anaerobic. Big difference. Now, what happens with the acid production? Well, in anaerobic glycolysis, you're going to have an increase of hydrogen, which of course decreases pH. You also have an increase of lactic acid, and lactic acid and your hydrogen ions are going to be transported out of the cell. And you have to have it buffered in the blood. So if you exceed the buffering capability of the blood, red blood cells, you're going to lead to lactic acidosis. Now, many cells, when they're dividing actively, are going to rely on anaerobic glycolysis. And production of lactic acid is normal. Lactic acid can also be found in sweat, and this can be antimicrobial. So what's important to keep in mind is lactic acid production is not a bad thing. It's a normal thing that your cells do. Your cells in one single tissue can be undergoing anaerobic and aerobic glycolysis, and a lot of it depends on where they are. How far are they from capillaries? The cells in your liver that are closest to the capillaries will get oxygen first. Those that are further away will have to wait to get oxygen, so they need to do anaerobic glycolysis to keep ATP levels up. Now, lactate is also going to be used in the Cori cycle. So this is an important cycle, especially during the fasting stage. And in the Cori cycle, you're going to have the red blood cell that can only do glycolysis. So this is only going to be anaerobic. They have no mitochondria. They have no way to do aerobic respiration. And so they're always going to generate lactate. They will release the lactate in the blood, and the liver will pick up the lactate through gluconeogenesis, make more glucose. The glucose will be released into the blood, picked up by the red blood cell, and you're going to keep this cycle going. Now your lactate are also going to be picked up by other cells, such as your heart. There are different forms of lactate dehydrogenase. We have our muscle, and we have our heart form, and other tissues have mixed versions of the lactate dehydrogenase. Now, lactic dehydrogenase is going to favor lactate, but if you have NADH, for example, then it's going to favor going to pyruvate. Now, lactic dehydrogenase is a tetramer, so there are 5 different tetramers. Your muscle and your heart have the same monomers that make up their tetramers. And then other tissues will have a mixture of muscle and heart versions of lactate dehydrogenase. You have your A subunits, which in your M, and your beta subunits in your heart. Now there are also other functions of glycolysis just to highlight here. So again, glucose is going to enter, and the glucose can end on pyruvate. Pyruvate can then go on into the mitochondria. So this is in the mitochondria. Or it can go to lactic acid. And this is in the cytosol. Or you can build on pyruvate. Pyruvate can actually form alanine. You have intermediates of your glycolysis that can form other products. 3-phosphoglycerate can form serine, and from serine you can get other amino acids formed. Glucose 6-phosphate can go through the pentose phosphate pathway and form your 5-carbon sugars, and these can go on to form DNA, RNA, and your nucleotides, ATP, GTP, etc. You also have glucose 6-phosphate can become glycerol phosphate, and this can make your fats. So this can make your tags or your phospholipids, etc. So there are many things that glucose, when it comes in, those carbons can do. They don't always have to make energy. But again, it depends on what your cell needs. If your cell needs energy, it's going to be used to make energy. Liver is going to be very important. In the fed state, it's going to synthesize fatty acids. And in the fasting state, it's going to synthesize glucose.